Breast cancer starts with tiny molecular abnormalities. But eventually those changes can reshape entire tissues. Breast cancer is the most commonly diagnosed cancer in women worldwide. Its incidence continues to rise globally. And despite major advances in therapy, it remains a leading cause of cancer-related death. So what actually increases breast cancer risk? Several factors contribute to breast cancer development. These include inherited mutations, aging, hormonal exposure, obesity, and lifestyle factors. Family history also plays a major role, especially in hereditary breast cancer. Now let's see how normal breast cells become cancerous. Breast carcinogenesis begins when DNA damage accumulates inside epithelial cells. Normally, repair systems correct these abnormalities. But when repair fails, abnormal cells begin surviving and proliferating uncontrollably. These molecular changes are strongly linked to key mutations. BRCA1 and BRCA2 genes normally repair damaged DNA. However, when mutations occur in BRCA1 and BRCA2 genes, genomic instability increases dramatically. That significantly raises breast cancer risk. But BRCA mutations are not the only drivers. Tumor suppressor gene TP53 normally prevents damaged cells from surviving. But mutated tumor suppressor gene TP53 allows abnormal cells to continue dividing. At the same time, PIK3CA gene mutations activate growth signaling pathways. So these mutations eventually create distinct tumor subtypes. Breast cancer is highly heterogeneous. Some tumors depend on hormones such as estrogen for growth and survival. Others overexpress human epidermal growth factor receptor 2. And triple-negative breast cancer lacks all major receptors, making treatment more difficult. Now let's examine the signaling pathways driving these cancers. In hormone-positive breast cancer, estrogen signaling drives tumor growth. Estrogen binds receptors inside cancer cells, activating genes involved in proliferation and survival. Another major pathway involves HER2. Human epidermal growth factor receptor 2 positive tumors contain amplified HER2 receptors on the cell surface. This continuously activates downstream signaling pathways, leading to aggressive tumor growth. But HER2 is not the only survival pathway. In cells, the PI3K KTMTOR pathway controls growth, metabolism, and survival. Many breast cancers activate this pathway abnormally, helping tumors resist cell death. Now, cancer cells also interact heavily with their surroundings. The tumor microenvironment strongly influences disease progression. Cancer cells communicate with fibroblasts, immune cells, and surrounding tissues. These interactions support growth and therapy resistance. Tumors also stimulate their own blood supply. As tumors grow, they require oxygen and nutrients. So cancer cells release angiogenic factors like VEGF. This stimulates new blood vessel formation around the tumor. Meanwhile, tumors also evade immune attack. Cancer cells develop ways to escape immune detection. One important mechanism involves PD-L1 expression. This suppresses T-cell activity, allowing tumors to survive. And eventually, some cancer cells metastasize. Metastasis is the deadliest stage of breast cancer. Cancer cells invade nearby tissue, enter circulation, and colonize distant organs. Bone and lung metastases are especially common. Another important aspect of breast cancer biology is tumor dormancy. Some disseminated cancer cells remain dormant for years before reactivating. There are also links between circadian rhythm disruption and tumor progression. So overall, breast cancer develops through a complex series of molecular and cellular changes that gradually drive tumor growth, survival, and metastasis.